In this review, we're going to focus on one section of Unit 6 of AP Bio, which is DNA replication. Before we begin, I've attached a couple videos I found helpful in studying for this unit in the description box, so make sure to check those videos out. In this video, we'll be covering DNA structure, DNA replication, the semi-conservative model, replication bubbles, and Okazaki fragments. Let's start by doing a quick review of DNA. DNA carries genetic information in every cell. A DNA strand consists of a bunch of nucleotides, and a nucleotide is a phosphate group, a sugar, and a nitrogenous base. They are connected through phosphodiester bonds, which form between the phosphate group and the sugar, and the bond comes from a dehydration synthesis reaction. DNA is a double helix, so there are two strands that wind around each other. They do so with complementary bases, and so guanine will correspond with cytosine, while adenine will correspond with thymine. These nitrogenous bases connect to each other via hydrogen bonds. Although RNA is one of the two strands of DNA, it's not the same, and it has several distinct differences. For one, in the sugar of the nucleotide, RNA has ribose, while DNA has deoxyribose. While this may seem small, and it kind of is, it just means that RNA has an OH, while deoxyribose has just an H. It loses an oxygen. Additionally, while the nitrogenous bases for DNA are adenine, guanine, cytosine, and thymine, RNA has adenine, guanine, cytosine, and uracil, replacing thymine. When we look at DNA, it does look like two strings, but they actually do have a direction. The ends of the DNA strand are either called a 5' or a 3' end. 5' has a phosphate at its end, while 3' has a hydroxyl group. In my head, I kind of remember it by thinking 3' plus OH, which is like two letters, adds to 5, and so it evens out. But that's just how I think of it. But like I was saying, both DNA strands have a direction, and they actually run anti-parallel. So while they are parallel, their directions are swapped. So 3' to 5' corresponds with 5' to 3'. This will be much more relevant when we actually get into DNA replication. With DNA replication, we're going to start off super broadly and then zoom in. To understand replication, you need to know the semi-conservative model. I'm simplifying heavily, but what this means is that when DNA replicates, it splits in half and serves as a template for free nucleotides to come and fill in and make two identical daughter molecules of DNA. So, like in this process, half of the parental DNA is conserved in each daughter molecule, making it semi-conservative. But in order for replication to even happen, DNA first has to untwist from its double helix to allow the nucleotides to enter. And it untwists at points called origins of replication. And these points essentially expand into bubbles that continue outward, allowing the nucleotides in, and they'll continue expanding until they fuse together, resulting in two daughter DNA molecules. But these bubbles don't just really pop up. They're the results of a lot of proteins and enzymes working together. So before we get into the specifics of the process, let's go over each enzyme individually. Helicase is the enzyme that unzips the DNA by breaking the hydrogen bonds between the strands. It's called helicase because hela refers to helix and ase refers to its enzyme. Single-strand binding protein, otherwise known as SSBs, are proteins that attach to parental DNA when it's being unwound to prevent the DNA from losing its structure or closing back together. Topoimerase sort of cinches the DNA on the outside of the replication bubble. It relieves the DNA that's about to be broken, relaxing it and allowing it to be uncoiled. Primase is the enzyme that synthesizes RNA primer. As the DNA uncoils, primase will continue to mark the beginning of the Okazaki fragments in the lagging strand. DNA polymerase III is the enzyme that runs along the parental DNA strand, reading the DNA and laying down the new nucleotides. It reads the parental strand from 3' to 5' and lays down the new strand from 5' to 3'. DNA polymerase III synthesizes the DNA on both the leading and the lagging strand. DNA polymerase I is an enzyme that goes over to where the primase placed the RNA primer and replaces the RNA with DNA. Ligase then connects the Okazaki fragments of the lagging strand by joining the new DNA to the actual strand. RNA primer, which we've already talked about, is the starting point for DNA polymerase III to start synthesizing in both the leading and the lagging strand. Before we really break this down, if this video has been helpful, please like and subscribe. Now let's put all these enzymes and proteins to work. In this diagram, we have an origin of replication expanding. Let's start with the leading strand first. Outside of the replication fork is topomerase, maintaining the DNA and relieving the tension until it's ready to be split. Helicase continues to unwind the DNA by breaking the hydrogen bonds, creating a replication fork, which is like the Y-shaped thing you see. Single-stranded binding proteins, or SSBs, attach to the now separated strands to prevent them from rejoining or losing their structure. The DNA polymerase III begins to synthesize a new strand from 5' to 3' continuously. Now, on the lagging strand, a similar process happens with some caveats that make it a little bit more difficult. Primase will place RNA primer on the lagging strand. DNA polymerase III, which reads from 3' to 5' on the parental strand, will start synthesizing at the RNA primer until it reaches another RNA primer on the origin of replication. As helices unzip the DNA, primase will go to the newly available DNA and place another RNA primer. Polymerase III will then go to that RNA primer and begin synthesis until it hits the RNA primer from before, signaling it to stop. Then, DNA polymerase I will go over the RNA primer and remove it and replace it with DNA. Lastly, ligase will join the Okazaki fragments together with the newly installed DNA by bonding the 3' end to the 5' end of the fragments, making a continuous strand. We covered all that. Let's review DNA replication as a process itself. Here I would pause the video and maybe write these down if you want to get a step-by-step notes. 1. Helicase unzips the double-stranded DNA by breaking the hydrogen bonds between base pairs, creating a replication fork. 2. Single-strand binding proteins, SSBs, attach to the separated strands to prevent them from rejoining. 3. Topomerase helps relieve the twisting tension ahead of the replication fork by cutting and rejoining the DNA strands. 4. DNA polymerase cannot start copying on its own, so primase lays down RNA primer on each strand to provide a starting point. 5. DNA polymerase III synthesizes from the RNA primer, adding new DNA nucleotides in the 5' to 3' direction. On the leading strand, this happens continuously, while on the lagging strand, DNA polymerase III produces Okazaki fragments. 6. DNA polymerase I removes the RNA primers and replaces them with DNA. 7. Ligase joins the Okazaki fragments together on the lagging strand, creating a continuous strand that happens on the other side as well. Thank you for making it to the end of the video. I hope that it was helpful. Don't forget to check out the other videos listed below. Maybe if you want, check out my other videos or shorts. And good luck on your test.